Nutrition in human beings. The food is first chewed in the mouth where it is mixed with saliva. This helps in proper digestion of the food. From the mouth, the food is taken to the stomach through the food pipe. Stomach has muscular walls which force digestive juices that aids in mixing the food thoroughly. Gastric glands present in the walls of stomach secrete hydrochloric acid, pepsin and mucus. The function of hydrochloric acid is to kill harmful microorganisms. Mucus protects inner lining of stomach from the action of acid. Pepsin helps in digestion of proteins. Food from the stomach enters into the small intestine where complete digestion of fats, proteins and carbohydrates takes place. Bile juice from the liver helps in emulsification of fats. Pancreas secrete pancreatic juice like trypsin and lipase for further digesting proteins and fats respectively. Intestinal juice secretes certain enzymes which convert proteins to amino acids, fats to fatty acids, and complex carbohydrates to simple carbohydrates. The inner lining of the small intestine has finger-like structures called villi which take absorbed food, whereas the undigested food is sent to large intestine from where it is removed. This is how nutrition is carried out in human beings. Activity. Aim: to study the digestive action of saliva on starch. Materials required: 3 test tubes, saliva, 2% starch solution, Benedict solution, iodine solution. Method: rinse mouth 2 to 3 times to remove any food particles and collect saliva in 3 test tubes labeled A, B and C. To B, add half the quantity of 2% starch solution. To C, add boiled saliva. Leave them for 5 minutes. Observation: at the end of 5 minutes, addition of iodine to A gives no blue color. Why? The action of salivary enzymes on starch converts it into sugar. Since there is no starch in the test tube, there is no blue color. Addition of Benedict's reagent to B gives red precipitate. Why? Benedict's reagent gives red color precipitate with sugar. In B also, the salivary enzyme has converted starch to sugar and the latter gives the red precipitate. Addition of iodine to one portion gives blue color, and addition of Benedict's reagent to the other portion does not produce any red precipitate. Why? C contained boiled saliva. Heating denatures the enzyme present in saliva. Hence, there is no action on the starch. The solution therefore gives blue-black color with iodine. Since sugar has not been formed, there is no red precipitate with Benedict's solution.